Hello, everyone. I'm delighted to be part of this conference dedicated to bioscience and healthcare. I would like first to extend my thanks to gentleman Dr. Daniel for his kind invitation to participate and present today a translational biology perspective using the yeast Saccharomyces cerevisiae. Translational biology has emerged, as you know, as a powerful paradigm in modern biological sciences, aiming to bridge the gap between fundamental discoveries and real-world application. So this is the science of conversion, transforming concepts and knowledge into tangible solutions. One of the most exciting area in this field is precision medicine, where the focus shifts from what we call one-size-fits-all treatment to tailored therapies based on the patient genetic profile as well as the environment and the lifestyle. In precision medicine, we speak of a transition from the bench to the bedside, where fundamental research guides clinical intervention. Pharmacogenomics, a key subfield, embodies this vision, this approach, by determining the most effective drug types and doses for each patient, again, based on their genetic and genomic profile. The ultimate goal here is to enhance treatment efficacy while minimizing side effects, making medicine more personal, more predictive, and proactive. Here you might wonder, what does a unicellular fungus have to do with personalized medicine and human disease? Here enters the Saccharomyces cerevisiae, the yeast Saccharomyces cerevisiae, an organism best known for its role in brewing and baking. But here we will focus on the yeast Saccharomyces cerevisiae as one of the most powerful model in eukaryotic molecular genetics. Let me share the slideshow first. Just it's taking 1 minute. Very well. I'm sorry for the delay. Okay, very well. And here the laser. I'm Fahd Nasr, a professor at the Lebanese University in Beirut, Lebanon. Here you will find the link to my personal website, which I invite you to visit. It features a range of courses related to translational biology. So please feel free to explore, use, and share them with your peers and students. The course web pages are password protected, but I am happy to grant you free access. Just send me a message using the email address shown here on the slide, and I will provide you with the necessary information to access. In addition, my website hosts over 200 publications covering various aspects of translational biology and beyond. In my presentation, I will provide some insights into how the yeast system serves as a powerful translational model, bridging the gap between fundamental research and therapeutic solutions. So why yeast as a model? Yeast has earned its reputation as an excellent model for several reasons. It shares fundamental cellular mechanism with human, including DNA replication, gene regulation, metabolism, signal transduction. It was the first, by the way, eukaryotic organism to have its entire genome, comprising 16 chromosomes, fully sequenced and published in 1996. It was a milestone that continues to support ongoing gene annotation efforts. Yeast is genetically tractable, meaning we can manipulate its genome with incredible precision. Using, for instance, homologous recombination, you can delete any gene within days. Further, you can conduct rapid and high throughput experiments and apply sophisticated genetic genomics and biochemical tools to study complex biological questions. It has a fast growth cycle, like a doubling every 90 minutes under optimal conditions, and it's cost effective. So yeast provide an efficient and scalable system to explore genetic interactions and molecular pathways relevant to human disease. So on this slide, you can see the budding yeast is beautiful. Saccharomyces cerevisiae with a small bud or daughter cell emerging from the mother, as you can see here, and separate at the end of mitosis, leaving behind a scar. The number of these scars indicate how many divisions the mother cell has undergone. These cells have a finite replicative lifespan and eventually age and die after a certain number of divisions that under good conditions could reach 30 to 40 division. I add here that yeast has also been extensively used as a model to study the mechanisms of aging. Furthermore, you should know that the scientific communities working on yeast is large. I mean, working on yeast as a model organism is large, reflecting its broad utility in biological research. However, Saccharomyces cerevisiae is not only a model system, but also a powerful biotechnological tool. It's considered as a grass organism. Yeast has been, for instance, widely used as bioreactor for the successful production of therapeutic molecule. Many medically significant proteins, such as HIV antigen envelope proteins, as well as hepatitis B, C virus proteins, epidermal growth factors, and alpha-1 antitrypsin, human growth factors, and many others. Now, yeast is equally a model for systems biology. In the context of yeast as a model for translational biology, systems biology allows or follows a dynamic cycle where new biological questions here drive the development of innovative technologies. And these technologies generate vast data sets requiring advanced computational tools and algorithms to extract meaningful insights. In turn, these insights refine our understanding of biological system, leading to new hypotheses, new discoveries, and yeast with its genetic tractability and extensive toolkit has been at the heart of this repeated cycle, leading to breakthroughs in disease modeling, drug discovery, and fundamental biology. But what makes yeast truly fascinating? It's the unexpected ability of the yeast to tackle the challenges beyond its modest evolutionary status. Yeast is ambitious. Despite being single-cell organism, it has proven invaluable in studying complex human disorder. It has been, for instance, instrumental in unraveling the mechanisms of neurodegenerative diseases such as Alzheimer's, as well as in cancer research, where it has provided groundbreaking insights into cell cycle regulations, oncogenic pathways, and drug resistance. AD as you know, is a major global health concern, with estimates, I believe, predicting over 100 million people who may suffer from AD by 2050. So the hallmark pathological features of AD, as you know, it's or include extracellular amyloid beta plaques and intracellular tangles of abnormal tau proteins. Tau proteins normally interacts with tubulin to stabilize microtubules and promote their assembly. However, when misfolded, tau can form insoluble aggregates contributing to neurodegeneration in AD and other related diseases. So yeast models have played a crucial role in advancing our understanding of AD pathology and are expected to contribute further to the development of targeted therapies and preventive strategies. The yeast system has proven to be an invaluable model as well for studying complex processes such as here on the list, you have eukaryotic cell cycles, signal transduction pathways, morphological switches, and gene expression regulation, among others. But surprisingly, it has provided insight into unexpected biological phenomena. Yes, yeast has been used to investigate fundamental evolutionary transition, such as the shift from unicellularity to multicellularity, one of the major leaps in biological complexity. The ability to model such transitions in a simple eukaryotic organism confirms the yeast adaptability as a research tool and its unique position in addressing fundamental and applied scientific questions. Listen, this is one of our experiments. In 1996, we have identified the CBK1 gene. Located on chromosome 14, it was a part of our effort to sequence the yeast genome. Inactivation of CBK1 leads to cell aggregation, as you can see down here, where cells remain physically attached and difficult to break. A quite frustrating phenotype, really difficult to handle. So as shown on this slide, CBK1 works upstream of ACE2. Now, just to make it story here, because it's quite complex, CBK1 is a kinase that activates ACE2, which in turn induces expression of many genes responsible for daughter cell separation, including those that degrade the bud-neck septum for the separation of the daughter cell of the bud at the end of mitosis. Mutation in CBK1 and ACE2 results in a phenotype that we call snowflake phenotype, as you can see here. A phenotype observed in other studies under different conditions. These snowflake yeast propagules exhibit characteristics of multicellular organism. In fact, they display a division of labor between apoptotic and reproductive cells. Look here, the red one. Some cells die to break propagules into smaller ones, while the reproductive cells generate more cells for growth. So in the time-lapse video I'm showing you here, you will observe the snowflake yeast propagules dividing to form new propagules. Something really amazing. Let me show you again. So the division of labors between cells within the snowflake here, propagules, indicate that some cells now are specialized to go through apoptosis and to break in order to generate new small propagules, while other cells at the periphery will divide to ensure the growth of the organism. And here you can see the time-lapse video again. It's amazing how the propagule is now acting as a multicellular organism. Very well. With this perspective in mind, now we can explore how yeast has shaped our understanding of cancer biology and continue to contribute to the development of precision medicine solutions. As you know, the landscape of biomedical sciences is all the time evolving. and the need for translational model has never been greater. We stand now at a crucial moment where fundamental discoveries must somehow swiftly transition into clinical applications. The complexity of human diseases, mainly cancer, demands models that are not only genetically tractable, but also robust enough to offer mechanistic insights and therapeutic strategies. Yeast has proven to be an ally in this endeavor. For instance, cancer is not a single disease, but a collection of disorders characterized by hallmarks such as uncontrolled proliferation, evasion of apoptosis, genomic instability, and metabolic reprogramming, and other features. Yeast has provided valuable, in fact, in providing insights into many of these processes, starting with the cell cycle and checkpoint. The discovery of cyclin and cyclin-dependent kinase, which regulate, as you know, the cell division, was largely driven by yeast genetics. Pioneering work on yeast cell cycle genes directly influenced cancer therapies targeting CDK. You should know about the palbociclib. Now it's an FDA approved for breast cancer treatment. For genomic integrity and DNA repair, yeast has been instrumental in uncovering DNA damage response mechanism, including homologous recombination and non-homologous end joining. The yeast RAD52 is famous. The yeast RAD52 pathway, for instance, has provided insight into BRCA1/2 related breast and ovarian cancers, paving the way for what we call PARP inhibitor-based therapies. You know, the inhibition of PARP or poly-ADP ribose polymerase in HR deficient tumors cause cell death of tumor cells while sparing normal cells. In protein homeostasis and aggregation, we know that misfolded protein accumulation is a hallmark of both neurodegenerative diseases and cancer. Yeast models of proteostasis have identified a protein, the chaperones more specifically, and autophagy related pathways that are now being explored as therapeutic targets for multiple malignancies. Look at this slide. It's one of the multiple screens in the toolkit of yeast, and we call synthetic lethality. The principle is simple. The principle of synthetic lethality is based on the idea that inactivating two specific genes here, the red and blue, together leads to loss of viability. Where inactivation of either one, as you can see here, the red or the blue, has no effect. In the previous example, you remember the genetic interaction between PARP, poly-ADP ribose polymerase, and BRCA gene can be described as synthetic lethal. This concept is particularly useful in drug-induced inhibition studies, where the growth of haploid or homozygous diploid deletion strains is monitored to reveal synthetic lethal interactions. Now, in chemical genetic interaction, a gene product represented here by the red circle, shown here to the right, is inactivated by a drug, and you can see the drug here, okay? The drug in pink square. So, a second gene, the blue one, though non-essential under normal condition, as you can see here, becomes critical in the presence of the drug. So the viability of the cell will depend on the blue gene. So deleting this second gene results in hypersensitivity to drug dose that would otherwise be non-lethal in wild type cell conditions. This is a beautiful screen of the yeast toolkit. Now, one of the most remarkable applications of yeast in translational biology, its role in drug discovery. Yeast-based high-throughput screening has enabled the identification of compounds that selectively target cancer cells. Because yeast shares many conserved drug targets with human cells, it serves as a powerful platform for testing chemotherapeutics, uncovering resistant mechanism, and also identifying novel drug synergy. For example, starting with the middle one, the synthetic lethality screen. You remember, yeah? This study led to the discovery that BRCA1 2 deficient cancer cells are highly sensitive to PARP inhibitors, a concept now applied in ovarian and breast cancer therapies. Kinase inhibitors, another example, is the use of yeast to study human oncogenic kinase BCR-ABL, which drive, as you know, the chronic myeloid leukemia. It was first characterized using yeast model, leading to the development of Clevac, another brand name. So a breakthrough therapy that revolutionized leukemia treatment. In metabolic reprogramming, cancer cells, as you know, exhibit altered metabolic states, often shifting towards fermentation rather than oxidative phosphorylation, the phenomenon that you know very well as the Warburg effect. Yeast, being a facultative anaerobe, has been used to model this metabolic reprogramming, aiding in the development of strategies to exploit tumor metabolism for therapy. In cancer biology, as you know, many key processes are controlled by protein-protein interactions, making them attractive therapeutic targets. Here you have another screen, another tool, the Yeast 2 hybrid screen or 2 hybrid system. It provides a powerful platform to look for small molecules that can disrupt or stabilize these interactions. Since many oncogenic pathways depend on abnormal protein-protein interactions, such as Micmacs in tumor progression or P53 interaction in DNA damage response, identifying compounds that interfere with this interaction could lead to novel cancer therapies. Here in yeast, as you can see, we can express an interacting a protein pair like X and Y, as you can see here in the middle, to the DNA binding and activation domain of a transcription factor that were separated to produce 2 fusion proteins. Now, if the interaction occurs between X and Y, a downstream reporter gene is activated, triggering a specific selectable response. It could be cell death, for instance, here in the expression of uracil 3 gene in the yeast in a medium containing 5-FOA, this will lead to cell death. However, if a compound disrupts the interaction, preventing X and Y from interacting, then the transcription factor will not be reconstructed. And as a consequence, transcriptional activation is prevented, allowing yeast cells to grow on this selected medium. This approach, by the way, enables high throughput screening of potential drugs that target crucial protein-protein interactions in cancer. So given the yeast's genetic tractability and cost effectiveness, it remains a valuable tool for early stage drug discovery before naturally moving to mammalian models. Cancer, as you know, is a moving target. Tumors evolve, acquire resistance, and adapt to therapeutic pressures. Yeast, despite its simplicity, has provided an experimental framework to study these adaptive processes. Yeast models have equally been instrumental, for instance, in understanding clonal evolution, also in identifying resistance mechanism, and in exploring epigenetic regulations. Here, for instance, and within this context, yeast has provided major insights into chromatin remodeling and histone modifications, including the SWI/SNF complex. We are getting to the end, so let me wrap up by saying that yeast isn't just a useful tool in translational biology. I would say it's a game changer. From helping us understand cancer at the molecular level, okay, To speeding up drug discovery, yeast proves that you don't need complexity to make a big impact. The strength of the yeast cells or the yeast system lies in its simplicity, giving us a powerful, easy to work with system to tackle some of the toughest challenges in human health. Turning basic discoveries into real treatment, which is dubbed translational, is never a straight path, but one thing is certain. Model organisms like yeast keep pushing science forward as we keep searching for better ways to fight cancer. This tiny, remember, this tiny eukaryotic organism remains one of our greatest allies in turning knowledge into life-saving solutions. And with this, I have come to the end of my presentation. Thank you for your attention. Wish you every success in all your endeavors.